Hello everybody, welcome to session 4 of this lab video series for cell and tissue engineering. So here we are a week later. Remember in session 3 we concluded with putting the cell-seeded gel constructs, agar gel constructs in the incubator for a week. Here we are a week later, and the very first step would be to remove the spent media out of these well plates. The goal of session 4 is to repeat the trypan blue live data assessment that we did in session 2, but this time we're doing it in 3D gel constructs. So we had 3 gels that are going to be prepared for each group for this 3D viability assessment with trypan blue. So here I am in the process of removing the old media. Also, keep in mind that these are gel constructs, and so the gel is still pretty firmly stuck at the bottom of the well plate. So the next step would, of course, then be to try to remove the gels from this well plate environment. To assist with that, I'm going to use the edge of a razor blade to gently lift the gel or the cellular gel constructs up from the bottom of the plate, as you can see I'm doing right here. All right, so now they are relatively loose. Again, you want to be careful with all of these steps. I'm also using a pipette to separate them from the bottom of the plate. And here I am lifting the gel construct up. All right, and I've managed to detach it from the bottom of the plate because it is fairly sticky, right? But also being careful because there are those valuable mesenchymal stem cells in there. So you can see here I've detached the gel. It's sitting on the lid of my well plate over there, as you can see. Right there. And so at this point, we want to be in a position to then expose sections by cutting the gel. Okay, and so we use a razor blade again to essentially cut up this gel into thinner sections. When we say thin, how thin do we mean? Well, typically, in order to have the cells visible under a microscope, a regular microscope, we would need the thickness to be in the order of about 150 micron to maybe up to about 300 micron. If it is too thick, you won't be able to see the cells clearly, so you may have to go thinner. One way you can measure the thickness of your sections that you cut here with the razor blade is to use a microcaliber, which will then show you what the exact thickness would be. Here we're sort of ballparking it, and if you've done this with sufficient experience, you sort of know how thin it has to be. Again, the thickness would be in the range of about 150 to 300 micron for a regular bright field microscope. Okay, so at this stage, I have cut it into those sections, roughly 150 micron sections, as you can see here. And I am then, at this stage, going to put them in its own well plate for the time being, as you can see I'm doing right here. Okay, and just for completeness here, I'm basically cutting this gel up into 4 representative sections. So basically, each gel construct that I have, I would cut it up into 4 sections that are 150 microns each. And I'm placing each of these 150 micron thick sections cut from a single gel sample into each of its individual well plates. So each section goes into its own well plate, and we have 4 sections, so we have 4 well plates filled with 1 section each. Okay, so there we go. And I believe I have, yes, and that's the last, I believe that's the last section. So that's the fourth section, and that's going into that final well on the corner over there. Okay, so now we've cut our gels into their representative size sections. Okay, and at this stage, again, each section is about 150 microns in thickness. Okay, at this stage, we would like to go ahead and initiate our viability assessment with trypan blue. So for that, of course, we will need to then add trypan blue for or to each of these cellular gel sections. Okay. So one important thing to keep in mind is that, again, it's important to know what the area of each of these sections are, because you want to be able to report the cell density per unit area. And of course, each section is very irregular. So when you look under the microscope, you want to make sure that you also are able to use the microscope software or any image processing software to measure the area within the region of interest. So you can report your cell in terms of a density, in terms of X number of cells per millimeter square, for example. All right, so now I'm getting ready to get the trypan blue out. We add 20 microliters of trypan blue to each gel section. Again, refer to your session 4 protocol for exact numbers, but 20 microliters is typically a standard for 150 micron thick, small-sized gel section like the ones that we cut up. And here you can see me adding the 20 microliters, pipetting that into the well plate, which contains each of our gel sections, and we cut up 4 such sections. Okay. We can, after we've added the trypan blue, which we're almost done here with each of the 4 sections, we would need to then incubate this in the incubator for about 5 minutes to allow the trypan blue to really get into contact within the gel construct and to basically interact with the cells. Again, as you remember from session 2, here I'm mixing the trypan blue in. As you recall, the live cells, they have no color, but the dead cells will light up blue because they start absorbing the trypan blue dye. Okay, so then you know which cells are dead, blue cells, and live cells have no color. So I mix the trypan blue into each of the 4 gel sections, each section being 150 microns thick. And again, at this stage, we would put it in the incubator for about 5 minutes to incubate. Following that, we could then look under a microscope to do our live and dead cell count. All right, so here we have our well plate with the cellular gel constructs already incubated for 5 minutes in a standard incubator. At this point, we have it under a microscope, and we can do our cell live and dead cell count using the trypan blue assessment, the trypan blue that was already mixed in. And we have these 4 irregularly shaped 150 micron thick gel sections. And so we want to be able to measure the areas of each of these irregularly shaped sections so we can report the area of these live dead counts. So sort of like if we had 10 live cells, we could say 10 live cells per square millimeter. If there are 3 dead cells, 3 dead cells per square millimeter, and so on. And then average those out across the 4 sections, and then we can average those out across the 3 samples per group. So here's a picture of what the live dead cells look like under a microscope. This concludes session 4.